you see any more coming in, Dr. Phelps, or good to go? All right, cool. Thank you everyone for joining us today. My name is Kieran Smith. I'm a first-year PhD student in the Conservation Sciences Department in Dr. Phelps' lab, recently admitted there. Today I'm very pleased to welcome Dr. Mikki McComb-Kobza. Mikki is one of the top shark experts in the world, and her work focuses on sensory biology and ecological physiology of shark skates and rays. She's a tireless advocate for sharks and shark habitat. She seeks to highlight their global population declines and frame new directions for their conservation. Mikki speaks, teaches, and conducts research around the world, including in South America, Africa, Asia, and Australia. Her outreach efforts have been covered by BBC, National Geographic, Discovery Channel Shark Week, and CBC National Radio Canada. She's really working to bridge the gap between research and public engagement. She believes that when people are aware, they care, and when they care, they act. She is currently the executive director of Ocean First Institute, and this allows her to promote research, conservation, and sustainability. The Institute also supports expeditions and a variety of education initiatives that reach students of all ages. Mikki holds a PhD in integrative biology from Florida Atlantic University. Woohoo! The author of numerous scientific publications. So with that, Mikki, thank you very much for joining us. I'll hand it off to you. Just, I guess one other thing is if you have any questions along the way, please feel free to post them in the chat. We'll do our best to get to all of those. Also, if you'd like to just at the end of the talk, we should have about 15 minutes. Feel free just to use your voice as well and ask Mikki directly. So yeah, Mikki, go ahead, take it over, and thanks again for joining us.
Okay. Well, hello, everybody. It's so great to be here. Thank you so much, Kira, for the invitation. Kira and I were in graduate school together, so it's so exciting to have an invitation to be here today. So I'm very grateful. I'm going to go ahead and share my screen and start my presentation. Okay, so Kira, can you still see and hear me? Okay, perfect. All right, well, thank you all again for the invitation and for being here today. My name is Dr. Mikki McComb-Kobza. I'm the director of Ocean First Institute. And today I'm excited to be here to share with you the story of sharks. And I'm going to be talking a little bit about how I got interested in sharks in the first place. And then I'll talk a little bit about their evolutionary history, their adaptations. I'll share some of the strange, weird, and wonderful species that are swimming in our ocean today with you. And then I'll talk a little bit about the research that I do on sharks and why. And it's going to be a kind of a winding road a little bit on this, but I've had an interesting career in shark biology and studying sharks, and it's been it's a great story to be able to share with you, to show you how I started in some of the physiological aspects and am now working a little bit more in the conservation. So I'm really excited to share that journey with you. So I'll go ahead and get started. And just to let you know a little bit more about me and how I got interested in sharks in the first place. I grew up in Colorado, so landlocked Colorado, had an amazing childhood out in nature. I came from those days where people were more feral and you could run and spend your childhood outside. And so it was a great start for me to engage in nature. But things changed for me when I was young, and I went and saw the movie Jaws. So for those of you that have seen this movie, you know what it's all about. It's about a big great white shark. And I went to the movie with my brother, and I left the movie terrified. I thought sharks were under my bed, under the kitchen table. I was terrified. So I had a shark problem. And the only way that I knew to overcome this problem was to read about sharks. My parents had this animal encyclopedia set. So I read all that I could about sharks, and I became fascinated. The story of the shark was captivating. And I realized very quickly that sharks were not the monsters that they're made out to be in the movies and on TV. And the movies just keep coming, right? And they're more and more ridiculous all the time. And so it really spurred for me an opportunity to share the truth about sharks and to really share their story. And so as I got a little bit older, I moved down to the Florida Keys to pursue a dream to become a scuba instructor. And that was an amazing time. I spent many hours underwater with sharks, learning more about their behaviors and just getting to be up close with them. And that was an incredible time in my life. And it opened up doors for me to go back to university. And I ended up going and getting a PhD, studying shark physiology and sensory biology, trying to understand how sharks find each other underwater, how they use their different sensory systems in combination. And so it was a really exciting time to learn more about sharks in the lab and to work along researchers all over the world. And then after doing a postdoc in Florida, I ended up moving back to Colorado, of all places, and started Ocean First Institute. And the nonprofit organization is all about research and education, all with the aim of conservation. So getting on now to sharks. When we talk about sharks, it's really important to understand one fundamental thing, and that is how long they have been around. And so our universe is thought to be over 14 billion years old, our planet Earth 4.6 billion years old. And as we look at it from space, we have a water planet, and life got really big and interesting around 440 million years ago. And this is when we saw the rise of the first jawed vertebrates, and that includes the sharks. And so from the fossil record, we see animals like Heliocoprion. Sharks' teeth are modified scales. And so we see these amazing evolutionary iterations like Heliocoprion that has this world tooth like saw in its jaw, and ones like Stethiocanthus that has what looks like an ironing board on its back full of bristles and bristles on top of its head. And those were seen only in the males. And so you have to wonder if that had anything to do with reproduction. So amazing for us to think about the fossil record of these animals. And of course, these are animals that were, here long before the dinosaurs, which we think of as ancient creatures. And we know dinosaurs are no longer here due to the asteroid impact, which changed conditions so much so on Earth that many species did not survive. But who was here swimming in the ocean at the time? It was the sharks. So the story of the shark is truly a story of survival. They have lived through 5 mass extinction events. And as we all know, we are living through the 6th right now. So sharks, skates, and rays are collectively known as the Elasmobranks, and they have a shared common characteristic. That is that they have internal skeletons just like you and I, but instead of bone, their skeletons are comprised of cartilage, so they are called the cartilaginous fishes. And because they have been on the planet so long, we have sharks and rays that have radiated into nearly every aquatic habitat on the planet. So we have sharks living on the brightly lit coral reefs. We have beautiful polka dot stingrays that live in the Amazon basin. I was able to go study these beautiful Matoro species. They're part of the ornamental aquaculture trade. We have sharks like this Greenland shark living under ice in the polar regions, and new research tells us this shark may live upwards of 400 years, making it one of the longest lived vertebrates. And then we have sharks like this frilled shark living in the deep dark benthos, where perhaps no sunlight penetrates. And imagine the adaptations for this animal to successfully survive. So when we study sharks, again, it's important to understand the tremendous diversity that we see in this group. There are over 500 living species swimming in the ocean today, all of which have different body shapes, different body colorations. Some sharks have to continually swim to survive, whereas others can lay on the seafloor and pump water over their gills. The reproductive modes observed in sharks are quite diverse, from egg laying to very advanced placental association. So we see a tremendous diversity in reproduction. So what I want to do now is just take you on a quick journey to share some of the most amazing sharks that you may be familiar with, and maybe these are new to you, but I think they're worth mentioning because they are just fantastic. So this is the whale shark. This is the biggest shark in the ocean. It's not a whale, it's a whale shark. And you can see here that has beautiful polka dot markings on its body. It's spending a lot of time near the surface. This is my son and I swimming right next to it. And you might think, why would you do that next to a 50-foot shark? Well, these are filter feeders. And so they are filtering out plankton, tiny plants and animals, plankton, all day long. And that's what's happening here. And you can see that the biggest bodies in the ocean are being built by these nearly microscopic plants and animals. So to go from the biggest to the smallest, this is the dwarf lantern shark. This is as big as it gets. It can fit into the palm of your hand. And amazingly, the velvet belly lantern shark actually bioluminesces. So it uses chemicals inside its body to create light. And it is thought that the belly of the velvet belly lantern shark matches the downwelling light. So this is a form of camouflage that the shark is using. Okay, going on, this is another amazing shark. This is the Mako shark. These are the cheetahs of the ocean. They have special adaptations in their eyes, and this is because of the speed. Mako sharks can go upwards of 50 miles an hour. And what you're looking at here is GoPro footage on the back of a boat. And those are baits that are trailing off in the background. And this Mako shark is easily coming up and getting these baits, putting it into its mouth and taking them off. So imagine, if you will, all that's happening here with this shark and its sensory systems. The eyes are paramount. And the Mako shark actually superheats its eyes to above ambient temperature. And it does this to increase the temporal resolution, or the speed at which the eye is processing visual information. And so this is one of the amazing adaptations that we see in the Mako shark to allow it to be able to grab those prey, very fast prey items. All right, this next shark is another really interesting one. This is the big Eye Thresher Shark, and it does have a big eye, you can see that. But the action in this shark is really in the tail. It has a heterocersal tail, so one lobe of the tail is much longer than the other, and it uses it as a weapon in order to stun schools of fish. So you can see here, it's an action we call tail smacking, and this allows the thresher to hit 2-3 fish at a time, spin around and grab those stunned fish very easily. So this is a physical adaptation to maximize the feeding success in this particular shark. So the tiger shark, just as we have tigers in the jungle, we have tigers in the sea. Tiger sharks have gorgeous markings on their bodies that you can see. They are the #2 most dangerous shark, according to the International Shark Attack File, and they are generalist feeders. So we have found the most bizarre things inside of the stomachs of tiger sharks, including cannonballs, entire suits of armor, wine bottles, fur coats, you name it. Tiger sharks will pretty much eat whatever they can put into their mouths. This next shark is really interesting. It's called the epaulette shark. It lives in northern Australia. The story here is that it can walk. So there really are land sharks, so to speak. So the epaulette shark uses its pectoral and pelvic fins in an action we call punting to be able to walk, literally walk on the reef during low tide. It then has access to small pools where fish are trapped during low tide, where it can go in and easily make a meal. New research has described 4 new walking shark species just last year. This is one of the last sharks that I'll share in this series here, but this is the Goblin Shark. So this is by far and away the ugliest of the shark species. It has this really long, pronounced rostrum. the nose full of sensors to allow it to be successful in its deep water habitat. You can see those teeth are really needle-like in their shape, and form follows function. So these teeth allow it to grab fish and other items in the deep sea. And you can see they're pointed inwards. So once you go in, you don't come out. But the weirdness doesn't end with the teeth of the goblin shark. The goblin shark has another mechanism in its adaptation. Its jaws are not fused to the skull. And so their jaws can actually protrude out and allow them to capture these more elusive deep water prey. And it is just incredible. So the goblin shark, you don't have to look much further than nature to find strange inspiration. So here it is in the goblin shark, really an amazing animal. So to have that really strange head shape like a unicorn, to then having a head stretched out, hammerheads. Hammerheads are probably my favorite of all the shark species, and they have a phenomenal story. This is the scalloped hammerhead. Scalloped hammerheads school together in large groups, and they have a social hierarchy within their schools. There are dominant and subordinate females. They're communicating with each other to a degree that we didn't really understand before. And we're really just starting to learn and unlock some of the mysteries of these amazing sharks. And what you might not know about hammerheads that is really fascinating is that they are the most recent evolved sharks in the ocean. So they are the newest additions in the ocean, and they have this very strange head shape. And it really begs the question of why. And what you might not also know about hammerheads is there are more than just one species. So we have a whole suite of hammerheads. And what's fascinating about them is they have a gradation of head expansion from moderate to what we see there in the bonnet head, to the one in the middle, which is the scalloped hammerhead you saw a moment ago, to flat out bizarre in the one on the bottom, which is called the winghead shark. And so we have this whole series of head shapes, and genetics tells us that the shark on the bottom was the first to appear, and the more derived shark is on top, which was totally counterintuitive to what I thought we would find. And so the largest head appeared first and has been speciating back towards more of a typical shape, I think, as they've radiated out. And so this is what that winghead shark looks like. They have a head shape that's about 50% of their total length. So they look like literal boomerangs almost. And so I had an opportunity to go to Darwin, Australia to work on these sharks. And I went in the monsoon season and went fishing for them. And all I caught were saltwater crocodiles. It was unbelievable. Did not find a winghead shark at all, but was able to secure a few at the local museum. So was able to do a little bit of research on these interesting sharks. You can see here, it's called the Chondrocranium. That's the skull, is stretched out, and all of the features of this shark have been displaced. Its eyes, its nares, all of the sensors in the head. So it's a really interesting model to ask questions about sensory function. So there's been a lot of different ideas and hypotheses put forward to explain the head shape of the hammerheads. And one of those is hydrodynamic lift. So the airplane that you see is called a canard aircraft, and canard have wings on the front and wings on the back. That's very similar to what we see in the hammerhead head, as well as its pectoral fins. So there is evidence that suggests that the hydrodynamic function is supported. There have been flume tests done to show that hammerhead heads are quite hydrodynamic, more so than pointed nose sharks. And they're also highly maneuverable. This is a great hammerhead, and their head shapes and their bodies are highly maneuverable. And that is another hypothesis that has been put forward. Another is that they use their heads to help them feed. And in particular, many of the hammerhead species eat stingray, and they've been observed anecdotally eating stingray wings and using their heads to help get purchase on that. However, not every hammerhead can eat stingrays because they're not big enough. So this is probably not one of the true evolutionary drivers of the head shape. But there is another idea, and that was one that I studied for my PhD, and that's the sensory function of the hammerheads. And I put that funny looking picture in there on purpose. You can see the eyes looking up, eyeballs rotate, they move in the hammerheads. But I was really interested in what has the change in the head shape done to the way these sharks encounter their world. And one of the very interesting things that I was excited about was the visual system. So what do these sharks see with their heads and their eyes pointing out straight on these big hammers? So shark vision was something that I studied, and sharks' eyes are surprisingly advanced. They rival our eyes in complexity and function. One of the questions that I was asking about shark vision is whether or not they can resolve color. How quickly do their eyes work? Recall we talked about the Mako Shark and how it's cruising at 50 to 60 miles an hour in contrast to a angel shark that may lay on the seafloor all day long. Their eyes have different requirements. And so the speed at which their eyes work are very different. And the other is the visual field. So that's the most fundamental element of the visual system. What do the animals see around their heads, and how does that help them perceive distance, depth, and things like that? So I was able to look at the visual fields of hammerheads, and I bet my bottom dollar that hammerheads couldn't see straight ahead because their eyes were sticking straight out and pointed sideways. And it just seems so maladaptive, but that was the question I was interested in. So again, sharks' eyes are incredibly complex. They're, again, the story of tremendous diversity with their pupils, which are mobile, so they can open and they can dilate and constrict their pupils to control light. They have different structures to control the amount of light in addition to the mobile pupil, like a nictitating membrane or a tapetum. So they're fascinating to study. Their eyes, again, are very similar to ours. However, they have a lens that is crystalline, like a marble, and that lens is moved away from the retinal layer to accommodate or focus, whereas we deform our lens. So there's some subtle distinctions, but again, very similar. So one of the questions that I was really interested in was, again, looking at whether or not sharks' eyes were matched to the habitat in which they live. And matched, I mean by their sensitivity. So the color sensitivity or spectral sensitivity of the eye. So I was able to do testing on 3 different species, a black-nosed shark, and then 2 hammerheads, a bonnethead and a scalloped hammerhead. And the interesting part of the story is that, you know, sharks come in different, they live and inhabit different areas that have different spectrum of light. And so we all know the spectrum of light, different wavelengths, red are longer wavelengths than blue, and that has consequence in the marine environment where the shorter wavelengths penetrate a little bit deeper. So in clear water, we'll see blue domination, whereas in coastal murky waters, we'll see more green dominant colorations. And so the question is, if you have sharks from different areas, perhaps open water versus coastal, will they have different sensitivities? So that was the question I was interested in. And so one of the other things that's interesting to think about too is contrast. So if sharks have a little bit of a visual offset, they'll be able to see in contrast things against a backdrop very well. And so contrast is another hypothesis that we were looking at. And then important to sharks, I'm sure you've probably heard, don't go swimming at night or dusk or dawn. And there's a reason for that. And one of the reasons is that we believe sharks' eyes are most sensitive to that transition time. And that is a highly variable time where fish and other marine life are changing from diurnal daytime to nighttime. There's a big transition, and so if you have a visual advantage, that literally can be a life or death advantage. And so we wanted to look at the sensitivity during twilight in these animals. So again, the full visible spectrum, you can see the day, moon, and starlight spectrum, and then that very tight time during twilight. And so again, we tested all of these different species, and we used a technique called an Electro Retinogram. So if you went to an eye doctor, they might possibly do this technique on you. It looks a little barbaric, but it basically, the eye is an electrical organ, and as you shine light on it, it creates an electrical response that goes down the optic nerve to the brain for processing. We can capture that with a probe on the eye. So it's not, it's just laying on a corneal layer, and we can record the response to light. And that's what an electroretinogram does. So that's the electroretinogram waveform. And that's what we use to deduce things like spectral sensitivity. So here's one of our sharks, and we're shining different wavelengths of light onto the eye. And here are those electroretinogram waveforms that we would get under different light wavelengths and different intensities. And based on those results, we can figure out what wavelengths the sharks are most sensitive to. And here's where it gets really interesting. What we found is that there were 2 distinct peaks in the sharks, and the 3 that we tested. And what we discovered with that is that the sharks that came from blue environments, which are the 2 on the top and bottom, and the one that came from a green environment in the middle, had a shift. So the ones on the blue environment had a blue sensitivity peak, and the other had a green sensitivity peak. But here again is where it really gets interesting. What does that mean? Does that mean that they have the ability to see color? What does it mean? Well, when we look at the different cones that we as humans have, we have red, green, blue. We think sharks only have one cone. And they have rods, which help them in low light conditions. So each of the sharks had a peak in blue, which represents the rods. They had a peak in the green. So we think sharks are actually what we call monochromats. They have just one cone. And so it's possible that many shark species are actually colorblind, which is a bit of a surprise. Or maybe not. It depends on how you look at it. If you have an environment that perhaps the energy required to maintain those cones isn't really beneficial to you, then evolutionarily you might lose it. And so we've seen that happen in pinnipeds. And so we also perhaps are seeing it in sharks. And so what's interesting is some rays do have multiple cones, and thereby, as we understand it, can process color, but the sharks remain unknown. And again, very few species have been tested, so it's really, we're not able to make blanket statements about sharks in general, but the handful that we've studied, many of them appear to be monochromats and not having that ability. So moving on to another really interesting question that I had, which was, can these hammerheads even see straight ahead? Do they know where they're going? How maladaptive would it be if they couldn't see straight ahead? So same technique I just mentioned, the electroretinogram, used to calculate the visual field around the head of the hammerheads. And what I found was so counterintuitive. That's why science is so amazing. You just get so many surprises. And what we found is that, whoops, I'm sorry, let me go back. We found about a 10-degree binocular overlap in normal sharks. As we looked at the first bonnet head, it got a little bigger. Then when we went into the scalloped hammerhead, we did have binocular overlaps, and they were 3 times bigger. And then when we looked at the wing head from Australia, it was nearly 50 degrees. And what does this mean? It just means that these sharks have overlaps in front of their heads, allowing them to have excellent depth perception. And so you can see here in this little model, this is what a hammerhead would see with its eyes. And again, the eyes move, the eyeballs rotate. And when they rotate, you can see here, this is what they see. So that double green triangle in front of their head, that's where that binocular overlap occurs. But you can see also this really big blind spot. So there is an area just anterior to the snout where they can't see. And again, what does that mean? They don't have hands. They've got to be able to put everything into their mouth. How do they do it? If they can't see, what do they do? And the blind spots we calculated are quite large, especially in the hammerheads, quite large. And so I started to think about other sensory systems that might be utilized for this void. And so sharks have the ability to detect weak electric fields. So every living thing gives off an electric field when it's underwater. And so sharks have evolved to detect it. And so if you see all these peppery dots on the underside of this bonnet head, those are jelly-filled canals called the ampullae of Lorenzini. And they allow the shark to literally sense living things that are giving off weak electric fields. And so I wanted to know, does that visual void, that blind area, do they use the electrosensory system to help them locate prey? Is that what's happening? And so we devised an experiment in a captive condition with bonnet head. So that's what's swimming there on the screen. We put down a weak electric generating device where we could flip a switch and create prey mimicking weak electric fields. And we used squid juice to get the sharks into kind of a hunting mode. And then we would flip the switch, the battery, basically, on the outside of the tank to create that weak electric field, and the sharks would come in and attack. So it was incredible. They were biting literally at sand, thinking that there was something there because their electrosensory system was being cued in. And again, the interesting part for me was, what's the distance? How far out does this go? And I was able to map that out for all of our species of interest. And what I found was, maybe predictable. I found there was 100% coverage of that visual blind spot with the shark's electrosensory system. So all it really is saying to us is that these sharks are incredibly well tuned for their environment, and these sensory systems are working in combination incredibly well. They're fine-tuned to help these sharks be the perfect predators that they are. So you might be thinking to yourself, okay, that's interesting, but how does this really help us? And at some point after the talk, you can talk to Keir. He worked really hard on some of the practical applications of sharks and electroreception, keeping sharks off of lines by using the secret communication channel with them. But one of the things that I discovered on my road in shark research was there was another elephant in the room, and that was that sharks are in global decline. And it's estimated anywhere from 40 to 100 million sharks are taken out of the ocean for shark fin soup. It's an Asian delicacy, and with an emerging middle class in China and Asia, it has pushed sharks to the brink of collapse in many populations. And so the hammerheads that I love and have been talking about for a while now are now critically endangered species. I have to have special permits to continue to work on them. And it's a sad story. And so I have refocused my efforts to a project down in Costa Rica in the Eastern Tropical Pacific, where we have seen dramatic declines. Up to 90% of scalloped hammerheads in this area have been removed, and sharks reproduce slowly and don't have a lot of young. And so the pressure on them is intense. An area in southern Costa Rica called Golfo Dulce, I was introduced to friends there who were noticing baby hammerhead pups in this bay, the Gulf, and they knew that fishermen were using them as bait, and they didn't understand that they were critically endangered species. And so I started working with them to try to understand the habitat use and why we were seeing so many little hammerheads. And so we started tagging and tracking the hammerheads to understand where they were going. So this is a little scalloped hammerhead, very, very young. We tag them, collect data on all of the morphometrics, and then we let them go. And so we have to take a moment just to make sure the animals are able to swim off strong. We don't want to let them go and have them sink to the bottom. So as soon as they're strong enough to swim off, we let them go. And that project, I'm really excited to share with you. In 2018, the Costa Rican president declared Golfo Dulce as the world's first hammerhead shark nursery. So it is a protected area, and it is allowing these hammerhead sharks refuge in order for these young hammerheads to grow up into adults, to really help bolster the population that really needs it right now. So what an exciting project that was. And we continue on doing work in Costa Rica. We're working now in an archipelago where we're tagging different species of interest, including the giant Pacific manta ray, which is incredible. And what we do with that information is we put in acoustic tags so that we can figure out the local movements, the fine scale movements of these animals. So this is a listening station. So this is an acoustic receiver that's underwater that will pick up the movement of these animals as they swim by. And so we're trying to, Costa Rica is a data poor country. And so for us to be able to start understanding where these animals are moving, the seasonality to it will really help us begin to protect them. And in a nearby area, we're also working on bull sharks. So this is a bull shark in Isla Murcielago. Bull sharks are the number 3 most dangerous shark, but these are amazing animals that move in and out of freshwater systems. And this is a national park where they're protected. But again, we find evidence of long lining and removal of these animals quite often. So we're very interested in seeing how these populations of bull sharks, these residential bull sharks, are doing. All right, I'm going to switch gears just a little bit here now and talk a little bit more about more recent research that I've been involved with. And that is with the white shark, which is the #1 most dangerous shark, according to the International Shark Attack File. And here you have a shark named Deep Blue, who was filmed off the coast of Guadalupe, off the Baja, Mexico coast. She is probably pushing the maximum length of great white sharks, which is about 22 feet. She is likely pregnant. It's hard to know, but female great white sharks can have upwards of 5 pups inside their body as they're pregnant, and those pups are over 5 feet in length. So you can imagine when you see her belly here, if she is pregnant, she's quite large. And so the question is, how large? And so I was asked to come to Isla Guadalupe to measure the sharks there and do it in a way that was non-invasive. So I had students help me devise a tool to measure the sharks underwater. And what we came up with is called, it's in the literature, it's called twin laser photogrammetry. And so essentially what you have, and this is used on terrestrial systems quite often, and it's also used in the marine environment. And what you do is you have 2 lasers that are set in parallel, a known distance apart, and you have a camera in the middle. And what you can do is shine those laser lights onto the side of the shark or an animal of interest as it goes by. And then what you can do is take that footage back, use image software to deduce the total length of the animal based on where those 2 dots were on the side of the animal. So it's a non-invasive and very effective way to gather data from these animals that are difficult to work with. So we were able to do that for 18 different adults in Guadalupe, which was really exciting. And we saw weird behaviors when we were there. This was a male great white giving us this head shake. And again, you know, great white sharks are the poster children of sharks, and we still don't have really a good understanding of some of the most basic behaviors of these animals. So there's still so much that we need to learn. And again, we don't know what this behavior is, whether it's territoriality, food in the water, females in the area. We're not really sure. And so one of the projects that I want to share now with you is one in the Northeast Atlantic, so up in Canada and Massachusetts, and also a little bit in the Carolinas. We're trying to determine white shark abundance and also their identification. And so we're dropping baited remote underwater camera systems. And so this is my student, and he's holding a crab trap. And on top of that trap, we have cameras mounted both ways. And we drop these cameras, and we set them down for maybe 3 hours or so, and then we retrieve them. And then we're able to see, what is down there. Do we have a lot of sharks? Very few sharks. When we get a look at the sharks, we can see if it's a male or female, what markings there are. So we get a lot of information from these camera drops. We also put in the traps acoustic receivers so that we can, if a shark is tagged and comes by, we'll detect it. We also use the lasers that you just saw to try to measure the sharks as they go by, and we've been successful with that. So this is the area that we're working out of, which is in Nova Scotia, Canada. And this is the vessel that we go out on. And here is just a quick video sharing what we do out there.
Program was out, which is basically baited underwater cameras. And sure enough, one of those bruvs had a beautiful female white shark swim right through the camera.
She really was interested in the bait. She was circling, giving it the look over. She started to bump the cage, give it a couple little bites, and it was exciting to look at it and try to imagine, what is she thinking? I think what's really exciting about this is that we're getting a glimpse into the life of these sharks, their movements, what time of year they're here, looking at the individuals, whether we have males or females, what are some of the markings that are on the bodies of these animals. So we're really getting a lot of information that we would never have topside by being able to have our eyes under the water.
Day 3 that we were able actually to attempt to capture and sample sharks, which was the 6th day of the expedition, turned out to be very productive. Even though a shark did not end up in the cradle getting the full suite of projects, we did validate that they were at that location, and we will want to get back there again.
And so this is kind of what it looks like and sounds like when we get this footage. This is in Massachusetts. This is a juvenile male white shark that came in and was very interested in our bait canister. And again, we can glean a lot of information from this. We can get sex markings. We even noticed scarring patterns. And so we're writing a paper on that right now. But it's really interesting what we learn from some of these underwater camera drops. One of the other things we do is we tag the sharks to try to understand their movements. We're really trying to understand juveniles and adults, males and females, how they're utilizing the Atlantic Ocean and where they're going and when they go, and perhaps even why they go. And so we use many different types of tags to help us understand some of these questions. And you can see here, this is a white shark, and on top of the dorsal fin on the top of the back is one of those satellite tags. And when the shark is swimming in the ocean and its dorsal fin comes out of the water, it sends a signal to the satellite, and then we can actually track where it's moving. And so I'm going to show you one last video here, just showing you some of the many things that we get when we bring these sharks in. We're getting blood, fecal samples for microplastics. We're looking at parasites. We're looking at their visual system. We're doing blood draws to get hormones and reproductive assays. So we're really doing a lot of different projects all at once.
We're here in beautiful Cape Breton, where the conditions were perfect when we first came out. The water's calm, the guys have their rigs out, and we're hopeful that we'll get a shark very soon. So within hours of starting fishing here at Nova Scotia 2020, we got our first shark. It was a big, beautiful male. Came on board 13 and a half feet long, about 1,400 pounds. We were able to measure him, put it in an acoustic tag, take parasites, take blood, and of course put our satellite tags on him before he left. And after he left, as he swam off beautifully, we decided to name him Breton for the people of Cape Breton, the great people of Nova Scotia.
So one of the things I'd like to just share with you as we come to a close here is just some of the data that we have from the satellite tagging has shown us some really extraordinary things. One of the last sharks we caught on that trip was Ngumi, which means grandmother in the local Nova Scotian indigenous language, the Mi'kmaq. And she was about a 50-year-old female. She was 17 feet long and probably weighs 3,500 pounds. So she was a big one. Just since we tagged her in this past October, she's moved down to the Carolinas. And I'll be going to the Carolinas in about a month to try to see if the sharks are there. We think that's where they're mating. And so we're going to go and have another 3-week expedition to try to see if we can find the sharks that are mating. But one of the other amazing things that we've learned from this tagging data came from another shark called Unamaki. We tagged her up in Nova Scotia the year before in 2019. She went all the way from Nova Scotia to Louisiana, back to Nova Scotia, back to Louisiana, and then back out into the North Atlantic. So it has been mind-blowing to see some of the movements of these animals that we didn't know before. And we're starting to see movements of these animals in groups. Again, we're seeing a lot of them congregating in the Carolinas right now, could be a mating area. Again, we're trying to really get our arms around what's happening with these animals and why they are where they are. That's really the end of the end of the study, is to try to understand that. So sharks are critical, as we all know, to the habitats. Really, the job for biologists, I think, now that are in conservation is to change the perceptions. And one of the ways that we do that is we take students with us that are younger and have them collect data on sharks and share the story of the sharks, share that in citizen science projects. And we take students on expeditions to tag sharks. So this is one of our students tagging a tiger shark out in Florida. And so it's I think really incumbent upon us to share our love of the natural world with young people, to get them to care and hopefully get into the natural sciences. So with that, I will stop sharing, and I would love to take questions if you have any.
Thank you so much, Mikki. That was super cool. Very exciting to see some of the research that you've done. We do have a few questions in the chat. I guess I first wanted just to start out. Does anybody want to just ask a question directly to Mikki? Figured I'd open that up first. Please feel free to take the floor, ask her a question directly. I'll give you a few, maybe 10 or 15 seconds to work on that one, and then otherwise I'll go into the chat.
Don't be shy. It's okay.
I can just ask my question. I put it in the chat, but I'll just jump on. Thank you so much for a great talk. I really enjoyed that. Very interesting to hear about. I'm really interested in your your research nonprofit work in general, and I'm curious if you have any I don't know if you could say a little bit more about how you decided to start it, and if you have any advice for students interested in conservation research at a nonprofit level, funding, things like that. Anything you could share. Thank you.
Sure, that's a whole nother talk. But you know what? I, as a PhD student, thought I was gunning for a research academic position. That was kind of what I wanted, what I thought I wanted to do, and always had a real love of outreach. And so as I ended my postdoc, I ended up moving back to Colorado, away from the water, and started the nonprofit organization, really trying to merge my love of research with my love of outreach. And I didn't know if it was going to work, and I didn't know if it was going to work from Colorado, of all places. But I had other people who shared my vision, and we started it. And the first few years was a lot of hard work and a lot of building up credibility and sharing what we were doing to get people to understand and to get funders to understand what we were trying to do, and that we were a good bet, a good investment. And we're 6 years in now, and I can say happily we are financially doing the best we've ever done. And we have an engaged board of directors, and we have amazing research interns. I do local conservation projects here in Colorado on fish restoration and aquaculture. So it's a lot of hats, it's a lot of work, but it's a great way to, I think, get other people on the train with you, so to speak, and to really feel like you're making a big difference. And for me, that was always at the heart of it. For me, was I wanted to look in the mirror and say, I'm doing something that matters. And that for me is what this job represents.
Thank you.
Sure. Thank you for the question.
I was going to ask a quick question. If you had any advice for students who are hoping to get involved with like shark conservation and research in any facet, kind of like what your thoughts would be on getting your feet wet in that regard.
Yeah, I do. I think, it's a funny as it is, it's a competitive field, and there are only a certain amount of labs that do the research. And my recommendation would be to find one of those labs, do your research, find someone that's doing something that really gets you jazzed and really gets you excited, and then read their literature, read their work, and contact them. Go out on a limb, write them an email, let them know you're interested in what they do, and perhaps think critically about how you might be able to do something in their lab that would benefit them and their research to take it one step further. And I think that goes a long way. And I think, having, that interest and having an idea sets you above so many of the other students that might not. So I would just say get specific and think about some things that you can do specifically to help that professor.
Thank you.
You're welcome.
Maybe I'll go to the chat now. So there's a question in there. It says, wow, this is so cool, which is a thought I had as well. What do these hierarchies look like in hammerheads? Are these based on resource access, food and mates? It says, thanks for such an amazing talk. And I think that's getting back to kind of like the head shape and some of those other dynamics you were speaking about.
Yeah, the hammerheads, the scalloped hammerheads are the ones that we see schooling. And there are, so in particular out in the Galapagos, there are people that go out and spend time on rebreathers. So they're quiet, they're not bubbles. The bubbles scare off the hammerheads. But those that do the rebreathers are able to spend time in that shark universe for a little while, and they're seeing hierarchies. So there are dominant females and subordinate females, and there are communication structures that are starting to appear when people are spending more and more time underwater. And one of the holy grail things about these animals is that we think they may be using that electrosensory system in a way we didn't think before, and that was navigating the Earth's magnetic lines. And so out in the Galapagos, there are seamounts, and we're seeing through tagging and tracking studies that many of these hammerheads are using these super highways, if you will. And how do they know? And so we're thinking that perhaps the electrosensory system, although a short range sense used in prey detection, could possibly be used in other ways that we don't even understand yet. So, there's so many mysteries with the hammerheads that are still needing investigation.
Cool. That's great information. Another question is, were you able to confirm the reason so many young hammerheads were present in the Gulf? Like, is that a pupping area, or is it due to some other, influence?
Yeah, it definitely is a hammerhead shark nursery area. And so there's several criteria that you have to have in order to have a designated nursery area. And so we see the females coming in specifically to pup in this area. It's kind of an enclosed area. It's seagrass. It's got all the good habitat for these young sharks. And when I was out there fishing, it was unbelievable. We were just bringing in one after another, after another, after another. And so, and and the locals knew this. And so finally, researchers were able to come in there and demonstrate through data and their hard, big research project for years. They were able to designate that as a true nursery ground. And then the Costa Rican president declared it as a sanctuary. So, really cool research resulting in direct protection of these animals. And again, 90% loss in the Eastern tropical Pacific. So they need a break. And, protecting the young is really one of the best strategies to help those populations rebound.
Cool. I guess I have one question just in regards to that. Like, how long do you think they're staying in this nursery ground area before kind of taking off? And then on top of that, like, is there any downside to recognizing this as a pupping ground? Have you seen like people going there to target them? There's a kind of, you see that with, in the Bahamas with shark feeding and things like that. People, they know where they're going to be, they're congregating in those areas and they can target them. So.
Yeah, for sure. And they definitely realized that that was a risk. And so one of the things that the group, the researchers that you saw there, they're called Mission Tiburon, they're actually training rangers. So the government is funding rangers to be out on the water to help protect these sharks. That's how dire the situation is. And that's how wonderful it is that the resources are being dedicated for that. But and to answer your first question, the sharks are staying in there for the first year or two, and then they're starting to move out into other areas. And then eventually there's this whole big triangle between the Galapagos, Mapelo, and Costa Rica. And so you can see where the adults are moving, even though it's a fragmented population of what it once was, we still are seeing those pretty predictable movements through tagged individuals. And so hopefully these young will replace and be the adults of tomorrow. That's the hope.
Very cool. Another question in the chat. This was an amazing talk. Thank you so much. What advice would you give an aspiring shark researcher that lives in a landlocked location as it relates to getting involved in graduate research? Is there anything aside from, direct contact that you could give someone a leg up in breaking into the shark world?
Yeah, never give up. That's my biggest advice. Always believe. People will tell you can't do it or you're crazy. Never, ever give up, and find that person, again, who can be a mentor. Find someone who's running a lab and contact them, and don't give up. Pester them. It's okay. Send a couple emails. They're busy people. It doesn't mean if they don't respond that they don't care. It just means they're busy. So be persistent. Don't give up, and do your research. Read their papers. Flatter them. Let them know you know what they're doing and that you want to be part of it. And I think that goes a really long way. And there's also serendipity and luck. You might be at the right place at the right time. That means get out there. Put yourself out there. Take risks. It's okay.
All good advice. So we just have a couple minutes left. I had, one, I guess maybe closing question, unless anyone else had any they wanted to ask, but what do you think some of the most important pathways are for shark conservation? Is this, I think you're doing some awesome work through education, but then also, is there reduced fishing pressure? What do you think is the best path forward for us, and how can we contribute to that?
Yeah, it's really a little heartbreaking. There was just a paper that was published in Nature last week that said that, sharks, are globally in decline, and, 50 to 70%, and it's worse than we thought. And so it's a sobering realization. And we are in the 6th mass extinction. I mean, we can't deny that reality. And so as biologists and people who care about nature, it's overwhelming. But I would say that, one of the most important things we can do is to, inform policy, to inform the public, to get people to care about nature, to get people to think about things beyond their own bubble. And it's hard, especially in a pandemic. But I think the pandemic has helped us all slow down a bit and think about our relationship to nature. And I'm hoping things like that will get more people to think critically about the value of an animal in nature. Does that animal have the right to survive? Do we go out there and support that? Do we allocate resources to keeping these animals alive? And my answer is absolutely. I think we just need more people on that on that train to really push this forward. And I think it's a multi-pronged approach. It's education, research, policymakers, advocates. It's everybody. We all have a part to play, and everybody has the skill and talent that they can use to push this forward. And that's what I would ask everybody to try to do.
Very cool. Well, we're right at 1 o'clock. Mikki, thank you so much. Really do appreciate you coming and telling us your story and about your research. Yeah, and want to thank everyone else for joining us today. Mikki, stay safe and healthy in Colorado.
Thank you.
And yeah, look forward to catching up with you again soon.
Okay, thank you everybody for tuning in. Have a wonderful rest of your weekend coming up. Thank you.
Thank
Bye.
you.
Bye, everyone.
Bye-bye.